Thank you, the organizers, particularly Dimple, Ranjith, and Mithun for inviting me here. And it's a very pleasant kind of occasion to share our work. It's a beautiful campus. I have been longing to come here for a while, and it's really good to share our work experience. So I am from Shahai Institute of Nuclear Physics, Kolkata, and we are working on epigenetic regulators with a class of proteins called chromatin readers. So in the complex genomic organization, the information needs to be decoded, and there are multiple factors that lead to epigenomic regulations. To make a very simple representation, these histone tails which has these modifications, have the writers which puts the marks, erasers which removes the marks, and we are working on the chromatin readers which are involved in this kind of recognition of these modifications. So very simply, what are chromatin readers? These are kind of a barcode readers, and they can actually understand and kind of regulate these epigenomic modifications. So in the nucleosomal context, these post-translational modification, acetylation, methylation, phosphorylation, even proteins like ubiquitin moieties are recognized and kind of translated by these chromatin reader class of proteins. So basically in our work, we are focusing on human disease and mostly in the cancer context. And we have in the epigenetic toolkit a class of family of proteins like ZMIND8, UBR7, transcription factor 19, speckle 110. These are some of the candidates, and we are looking in the different cancer context. For today's talk, I'll be focusing on the breast cancer aspect. And what are the kind of questions we are looking at? How the gene expression regulations are being fine-tuned by these readers in cancer cells. We are also looking at some of the other different problems, like how the cancer cells extracellular matrix is contributing to the tumor cell niche, and also the invasive and metastatic potential of the tumors, and also metabolic reprogramming, and how these epigenetic regulators play a seminal role in that. So I'll take you to the first part of the talk. The first is the metabolic regulation and the role of the readers. So this is a tumor kind of three-dimensional representation, and it is needless to say that the chromatin domains in the core and the core of the tumor versus the different regions of the peripheral cells are very different. So there are different metabolic programs that are actually reinstated in the core of the tumor versus the periphery of the tumor. So as you can see, the core of the tumor is an oxygen depleted, nutrient depleted center. It's a hypoxic environment, and the glucose is basically converted to pyruvate, and then there's an axis which shifts it more towards lactate. So we are actually working on this kind of metabolic shift that is happening in the hypoxic tumor core environment, which is contributing to the proliferative potential, chemotherapy resistance, and other properties of the cancer cells. So we are working on this factor ZMIND8 for the last couple of years, and there are different school of thoughts which kind of projects this zinc finger type of protein. So there are roles and models that are established it as a tumor suppressor protein. So it could be a kind of modulus, kind of ATRA-mediated regulation that has been well studied, and also other interacting partner switch mechanisms are at place. Shows that it binds to KDM family proteins, and in different contexts it can promote, it can inhibit proliferation, oncogenesis, and the metastatic potential. On the other hand, there are groups which have started working on the tumor promotion models, and there is a paper that shows that in the hypoxic environment, the hypoxia response element sites, which are recognized by HIF1 alpha, this recruits the D8 containing complex, and it promotes angiogenesis, cell motility. So we are asking the question, moving from 2D to 3D context, how this ZMIND8 is involved in tumorigenicity regulation. So we started our experiments very simply from the clinics. We dissected the whole tumor models in the core region and the peripheral region, which are different molecularly, and this is the surgeons who can do this dissection and clinically give us these samples. And now when we looked into this tumor periphery versus core, we were surprised to see the ZMIND8 is one of the factors which is quite heightened in the tumor core. So indicates that particular microenvironment that is there is being fine-tuned by this ZMIND8 protein. So we moved ahead and established the 3D spheroid models in the lab, and you can see in this spheroid context, we first used the molecular markers like Ki67 and CA9 to establish the periphery and the core of the tumor. And then if you look at the ZMIND8 expression, it is again mostly confined in the tumor core region. So molecularly, something is going on with this epigenetic regulator in the tumor cores. So now we started mimicking this environment of the kind of core-like conditions by two ways. One is initially we started with low glucose and cobalt chloride treatment to activate the HIF1 alpha, and then later on we moved to this hypoxia chamber-mediated hypoxia activation, and you can see in HIF1 alpha activation quite robustly happening. What happens to ZM/ND8? So you can again see in both the conditions there is aberrant, there's a significant overexpression of ZMIND8 protein, so indicating it's a hypoxia sensor. So we did RNA sequencing analysis, knocking down ZMIND8, and out of the several biological pathways that has come up, one of the areas that we have focused on is the metabolic regulation aspect. So then we went ahead and performed chip sequencing from the core cells of the tumor, and you can see here they have the general recruitment in the promoter TSS, and again metabolic processes came up as major biological pathways. And again, very interestingly, when we looked at the kind of pathways through gene set enrichment analysis, we find a lot of genes are involved in the glucose metabolism regulation, including the hypoxia-like conditions that are involved here, glycolysis, gluconeogenesis pathways, and several other factors that kind of corroborates with the glucose metabolic changes that is happening in the tumor core. So we went ahead and performed immunohistochemistry analysis from these different tumors, and you can see the ZM/ND8 and HIF1 alpha has a beautiful correlation in these tumors in the context of expression. We also saw that in the tumor core, ZM/ND8 and HIF1 alpha does physically interact, and there are many target enzymes that ZM/ND8 regulates. Like in hypoxia condition, you see ZM/ND8 being elevated along with HIF1 alpha, and it leads to activation of LDHA and hexokinase 2. We again connected with our ChIP-seq data, and you can see HIF1 alpha and D8 being co-recruited in these important metabolic enzymes like LDHA, HK2 genes. So indicates that it's a co-recruitment model where both of these factors get recruited together and is regulating the glucose flux. So getting into the glucose flux, the first thing we did is looked into the extracellular acidification rate in the core tumor cells where we knocked down D8, and you can see there is a downregulation of the ECART as kind of expected. And we also saw that if you target ZM/ND8 in these cells, the LDHA, which is the master regulator to pyruvate to lactate transfer, this expression is highly downregulated. So this, along with this, we also measured the lactate pools in the cells. So these are the lactate pools, which shows again significant reduction when you target D8. And I haven't shown here, but acetyl-CoA levels show the reciprocal, that is where the core condition you target D8, these lactate levels are downregulated, acetyl-CoA levels get upregulated. So we finally went ahead and performed the PBMC invasion assays, where we take the PBMC cells and from the different conditions, the norpoxia where D8 is knocked down versus hypoxia again where the knockdown is done, we add these kind of cell media and look at the effect of the PBMC invasion, and you can see here this is very specific to the hypoxia context, where in the hypoxia you have a low level of invasion, but when you knock down this ZM/ND8, there is a heightened invasion. So bringing the things into a simplistic model, which we are doing, and so you can see that glucose to pyruvate and pyruvate to lactate transition in this ZMIND8 is playing a seminal role and diverting the metabolic flux towards the lactate. There is more lactate release, and then that can kind of suppresses the immune cell calling, and you can see that this is really a factor in the tumorigenic context. It is an oncogene in the context of the hypoxic tumor environment. Okay, so this is the part one of the story, and then I would take you to the second aspect, which I have mentioned, is extracellular matrix remodeling and how the factors epigenetic readers play an important role. So this is a quick introduction of the normal extracellular matrix and stiffened matrix in cancer. So what you see is there are lots of changes that happens in the matrix level, like collagen bundling and cross-linking and other things that really lead to stiffness of these extracellular matrix in the cancer. So these stiffened matrices have multitudinal functions that it kind of evades the growth of tumor suppressors, resists cell death, escapes immune response, acts as a drug-deficient barrier, and induces angiogenesis. For today's talk, I'll be focusing on this promotion of the invasion metastasis, although we are doing the respective other functions and the role of the ECM in this context as well. So this is kind of the ECM deposition and ECM alterations that happens. So the first phase in the cancer cells is ECM deposition, followed by post-translational modification leading to ECM cross-linking. That is very important. And finally, the ECM degradation, which is connected with this angiogenesis phenomena. And if you look at the cancer, it's kind of, we kind of speculate that we could actually bookmark the cancer, whether it's in carcinoma in situ, the basement membrane breakage and invasive tumor context with this ECM stiffness and rigidity and the context of the ECMs that we are studying. So we have been working with one important protein called E3 ubiquitin ligase UBR7, and we initially had showed that this UBR7, although it's a chromatin reader family, has acquired different function. It's an atypical PhD finger containing protein. It acts now as a H2B lysine 120 ubiquitinating enzyme. And we found that if you target the UBR7 in the cells, lysine 120 ubiquitination dramatically drops. And this has direct consequence in the cellular invasiveness, alteration in the EMT programs in the cells. With the ECAD, the epithelial markers gets down regulated, where the NCAD or Vimentin gets turned on. And in the animal model experiments, you can see that UBR7 wild type overexpressed tumors are kind of, it is a suppressor and the tumors do not come up so easily. We also looked at the patient level, the primary versus metastatic tumors, and see that there is a downregulation of UBR7 expression in the metastatic tumor context. So now why suddenly ECM thing came into our interest area is because when we took the triple negative breast cancer cells and targeted UBR7, we observed that there are multiple processes that get deregulated, but one of the fundamental process that was found to be differentially altered is extracellular matrix organization. So this was kind of very interesting to us. And again, if we kind of do functional annotation to the pathways and groups, we find the TGF beta signaling cascade was definitely affected when you target UBR7. So now, what we observed is we did unbiased targeting and tried looking at the different epigenetic modifications. So it was a H2B lysine 120 ubiquitinating enzyme. However, in the TNBC context, in the endogenous factor being targeted, we did not see an appreciable change in the K120 ubiquitination. In contrast, we observed histone H3 lysine 27 trimethylation, which was globally affected when we targeted UBR7. And following this, when we went ahead and did chip sequencing analysis, again you can see the 27Me3 levels were globally downregulated, affecting several genes and pathways which affect ECM. So this is shown for simplicity the TGF beta 1 gene, and you can see that when you do not have UBR7, the 27 trimethylation landscape is hugely downregulated, indicating that UBR7 is involved in actually regulating this ECM gene through some upstream pathway. So we performed IPMS study, and we took normal proliferating cells, MCF10A, and triple negative breast cancer cells, MDMB231, and we identified the interactome. And one of the particular complexes that hit our attention was this PRC2 complex, EZH2, EED2, and SUS12, while UBR7 did not interact with these protein complexes in the context of MCF10A. In the MDMB231 cells, we could actually get this entire complex protein isolated. So this was very interesting to us, and so this kind of get the model that we were thinking about is in the TGF beta gene promoter, UBR7 is definitely there, and it is instrumental in recruiting the PRC complex. So if you do not have UBR7 in the system, the ED2, SUS12, EZH2, all these subunits actually get dislodged. So we now hypothesize that UBR7 could be a formative complex component which is very crucial for the PRC2 recruitment to chromatin. And you can see here that if you target UBR7, the EZH2 recruitment through chip assays was dramatically compromised. And so if you do not have UBR7 along with PRCs, TGF beta activation happens, leading to activation of phosphosmad cascade, which turns on these ECM genes, collagen, fibronectin locks, and that is the signaling mechanism that we actually established. So now if you move from an adherent cell to a 3D model and you have UBR7 removed from the system, you can see fibronectin actually getting kind of hyperexpressed along with the LOX enzymes, which are the cross-linking enzymes. So these are the sphere formation assays, and you can see the total collagen content like that. We estimated fibronectin and other factors of the ECM. You can see there's an upregulation of these, the staining of these in the 3D context. showing the ECM is more robustly forming when you do not have UBR7 in the system. So from the tumor spheroids, we now estimated the LOX activity assays, and so you can see from the spheroidal cells as well as from the spheroid supernatant, you can see targeting UBR7 leads to an activation of the LOX activities quite robustly. And LOX, as I mentioned, is the enzyme which cross-links collagen and makes the ECM bundling more stronger and kind of more rigid. So now we went ahead to the mouse models and we targeted UBR7 and again complemented back with the wild type protein. And you can see the LOX activity as well as the total collagen content shows a differential expression when you don't have UBR7 in the system versus when you complement again wild type, it again reduces. So from the cells into the animal model, this actually works. And so these are the type 1 collagen stained tissues that you can see the similar way with the increase in the expression of the UBR7, of the knocking down of UBR7, the expression of this type 1 collagen fiber was distinctly increased in this context as you can see from the images. Then we went ahead to look into the patient samples and we did total collagen assay as well as LOX activity assays, and these are some of the kind of studies which clearly shows a kind of anti-correlation where UBR7 expression and LOX activity or total collagen content was inversely related. So these are the tumor microarray sections and showing some of these slides. You can see the kind of anti-correlation I'm talking about. Expression of UBR7 was much low and then fibronectin and collagen was heightened in the tissues. And so there was a stage grade correlation. This was more prominent in the higher grades of tumor that you can see this kind of anti-correlation functional. So finally, the context of matrix stiffening that we are talking about, we wanted to understand it in the cellular context, how we can score these assays. And one of the best ways to study the stiffness is doing atomic force microscopy. So here we cultured cells and then we looked at the native matrix formation, and the stiffness of these matrix was actually measured by AFM. So from the results you can see here, the stiffness was measured through DMT modulus, and you can see there is at least around 3 times the increase in the cellular matrix stiffness when you just do not have one factor UBR7 from these cells. So these questions could be all in vitro and might be all kind of some kind of data which we might not extrapolate in the cells, from the cells to the actual tumors. So shown here is the mouse tumors, and we again repeated our experiments where we did from the tumors the AFM studies, and you can see matrix stiffness is quite pronounced when you have this ECM overexpressed, and when you again co-overexpress ECM, the UBR7 along with these ECM proteins, you will see that there is a downregulation of this Young's modulus. So you can actually step it up, look into the animal model tumors, and you can actually measure the stiffness, which is beautiful, connecting it with the cell level data. Another small thing that I'll show here, but we are still working on this on much greater details. So we are looking at the cell-free DNA release from these matrices. So we know the stiffer the matrix, there's a release of the cfDNA is directly related to that. And so there are multiple mechanisms that are there in place, passive release models as well as active release models. We are looking into the active release models, which are extracellular vesicle mediated cfDNA release. These are some of the initial experiments, and we have done it in the mouse models also. And so what you see is control versus ECM overexpressed condition. cfDNA release is much higher, and again you have co-overexpression, you can reduce it back. So this cfDNA release, people say, is some kind of intermediate step which decides the metastatic potential of the tumor as such. So to understand that, we looked at the invasiveness of the tumor. So these are tumor invasion assays, and you can see that LOX overexpression, the invasiveness increases. Again, co-overexpression with that, we see that there is a downregulation. So that we can connect the dots and say that there is a direct regulatory role of UBR7 in the context of matrix stiffening and downstream the invasive functions that you can see. So a part of the study that I have talked about is just published, and you can see here the TGF-beta is a master regulator, how it is co-regulated by UBR7 and PRC complex, and that kind of suppresses the activation of the ECM genes. In absence of UBR7, the PRC complex recruitment is not happening, leading to TGF-beta activation, and downstream there is activation of these ECM factors, which are involved in directly leading to the alteration of the matrix stiffness of the cancer cells. So yeah, that's all. So these are my grad students, and some of them were involved in ECM projects, some of the metabolic regulation project, and these are my collaborators without whose help and continuous support, some of the experiments which are very challenging would have been difficult. And this is my project funding, and thank you for your attention.
Thank you. Questions?
Very nice talk. Thank you. I just have a question about CMYND8. And like a few years back, it was shown by Michaela de Virgilio's lab that it loads onto enhancers and it has a role in transferring RNA polymerase from enhancers to promoters. So if you take it out, you get polymerases more loaded on enhancers and promoters. Do you see that in your studies in context of enhancers surrounding the genes that you are studying?
Perfect question. Like there's a super enhancer activation model that those group has shown, and since we have the chip sequencing data, we actually see in the kind of cancer context, particularly the core tumor context, there are lots and lots of non-coding RNAs and the enhancer-mediated regulation which are coming up. So, but it's like a dots in the puzzle. We have to connect it and try to see how this entire thing fits into the magnanimity of the cancer. But yes, it's a very important happening event, and non-coding RNA regulation is in the forefront in this context.
Thank you.
Regarding the first part of your talk, I was curious if, because there is also metabolic changes, like the level of acetyl-CoA, I guess, are changing. So, and of course, this is very important for gene activation. So do you see crosstalk between the two, and how is it working?
That's a very wonderful question. So acetyl-CoA and the lactyl pools are kind of inversely correlated, and this guy is majorly involved in the control condition regulating acetyl-CoA, and in the kind of hypoxic condition regulating the lactyl-CoA levels. And that we hypothesize kind of has an important condition in the acetylation, lactylation of histones. We are trying to work more on that regards, and how this guy can switch the windows and promotes and that transfers the gene expression. But it is something to think about, that acetylation as well as lactylation of histones, both are transcription activation kind of context, but it's very context dependent event, early phase versus late phase. So it's all time dependent in kind of dynamics that is going on, something which we are very much interested in, and we are exploring that at the present moment.
Hi, nice talk. For the first part of your story, is it only specific for ER-positive breast cancer cells?
Okay, so we have been looking at the different etiology of the disease, like TNBC, we have one set of study, and the one that metabolic regulation stuff that we are doing is in ER-positive tumors, because of the fact that is more happening in the population, and the ER-positive tumors and HIF1 alpha correlation is very nicely can be done. So the complex recruitment onto many genes and in cancer enhancer recruitment models that are there in place with us now. So that shows that in ER-positive tumors, this is a very important event.
Is there any sequence specificity for the
ZMIND8? So see, it's a PBP containing transcription factor like PWWP, Bromo, and PHD domain. So together it gets recruited to certain epigenetic sites, certain histone PTMs it prefers. But saying that, there are certain DNA sequences inherent in the chromatin where it goes and binds. So it's not that everywhere the chromatin marks are there, it will go and bind. So there is a contribution of the DNA. That's why the enhancer activation model that we are proposing is there.
Thank you. Any other questions? If not, let's thank Chandrima for the nice talk.
Thank you.